Hello students, welcome to my class. I'm Keerti and today we are going to have a session on the topic DNA replication and protein synthesis. So this chapter comes under the title Molecular Basis of Inheritance. So before starting, let's decode the title of this chapter. This is saying molecular basis. It means something is going on at the molecular level. Okay, something that we are not able to see through our naked eyes. The molecules which are involved in this process are or cannot be seen. This is happening at the cell level, right? So, all the processes that we are going to discuss in this chapter is happening at atomic level. Okay, so let's start with our chapter. This chapter explores the structure and function of DNA. As you already know, in humans, DNA is the genetic material. So, this chapter concerns with DNA replication, how this genetic information is going to express through protein synthesis. Right? DNA, the full form of DNA is deoxyribonucleic acid. Right? And in most of the organism, it acts as the genetic material. For example, in humans, some organism may have RNA as genetic material, that is ribonucleic acid. But in this chapter, we are concerned with our system, so we are going to study about how DNA is replicated, how DNA is transcribed, and how ultimately the protein synthesis is taking place. So, DNA stores the genetic information in the sequence of nucleotides. What is this nucleotides? This nucleotide is made up of nitrogen base, sugar, and a phosphate bond. Right? And these DNA, it directs the synthesis of RNA and proteins through the specific process that are known as transcription and translation. I'm going to study in this chapter. Right? See this structure. This is DNA. Okay. So, as I already told you, that this DNA is made up of nitrogenous base, sugar, and a phosphate group. These nitrogen bases are of 5 types, right? Adenine, guanine, cytosine, thymine. These are present in DNA. While this thymine is being replaced by uracil, which is written as U in case of RNA. Rest is same. Okay, so RNA will have adenine, guanine, cytosine, and uracil, while the DNA will have adenine, guanine, cytosine, and thymine, right? So this backbone of the DNA, the structure, is made up of these nucleotides that are connected. These nucleotides, these nitrogen bases are attached with sugar and phosphate. Okay. So we already know, we have learned in our previous chapter that G and C are joined through triple bond, right? And adenine and thymine are joined by double bond. So, so this is the basic information about the DNA. This DNA occur as a double-stranded helix with complementary base pairing, right? That occurs between adenine and thymine, that I already told you, and guanine and cytosine, right? The strands are anti-parallel and coiled in right-handed fashion. What does anti-parallel means? It means if this double-stranded DNA, one is running from up to down, then another is running from down to up. Okay? This is written as 5 prime to 3 prime. And since these strands of DNA are complementary, are parallel to each other, so another strand will be named as 3 prime to 5 prime, right? And in these structures, these adenine, guanine, thymine, cytosine are present, and they will bond to their respective counterparts, right? It means that if adenine is present, it will bind with thymine. If guanine is present, it will bind with cytosine. If thymine is present, it will again bind with adenine. So these are anti-parallel, and this is what we call the complementary base pairing, right? In case of eukaryotes, this DNA is wrapped around the histone proteins, right? Which you might have read in class 11. In this chapter, we are going to study how the DNA is replicating. Okay, so you must have a basic idea about the structure of the DNA. See, these are the histone proteins. Histone are octamer in nature, and the DNA will be called around that histone protein. Okay, one structure of this is known as nucleosomes. This nucleosome, in n number of times, it will coil and form a very condensed structure that is known as chromosome. So, if this strand of chromosome, we are going to extend it, it will look like this only. Okay. And if this DNA will also be coiled open in the next step, this DNA will look like this. It's double-stranded DNA with complementary base pairing, right? So this is a typical diagram showing the presence of histones, the DNA, and the structure of how this DNA is going to coil and ultimately forms a chromosome, right? So how this DNA is replicated? See, this DNA replication follows complex process which we will study when we will go into deep in this chapter. This session is all about giving you an overview that what amount or what topics are there in this DNA replication and transcription processes, right? The information in the molecular biology always follow this central dogma that it is the channel of passing information one way, right? It means that this DNA will contain some kind of information. It will travel through the formation of RNA, and this RNA will ultimately lead to the formation of protein, right? So, for this process, different steps are required, different kind of enzymes are required, different kind of uh, catalyzation type of component is required, right? So this DNA will replicate, and when this replication is done, replication means nothing but creating copy of this DNA, right? This DNA, using the information of this DNA, RNA will be formed. And when this information that RNA carries is transcribed, this will lead to the formation of protein, right? So in DNA replication, DNA makes identical copy of itself before cell division. We already know that cell division takes place through the process of mitosis and meiosis, right? So if a single cell is going to divide into two cells, it must replicate itself. It must create an exact copy of itself. So for this purpose, all the information must get duplicated and then get transferred into the upcoming cells. Okay, this ensures what? This ensures the genetic continuity from one generation to the next, right? Like the humans are reproducing and producing the organism of their own kind, right? We cannot see it. It never happens that a cow is going to give birth to a human child, right? A human child will only give birth to a human parent will always, or human male and female, the female will always give birth to a human child, right? So that happens because this DNA contains all set of information that is needed to produce an organism, right? So in this process, DNA is replicating and generating a copy of itself, right? This DNA replication is also known as semi-conservative replication. See, this process of DNA replication, we will deal later on, that how many enzymes are involved, how this double-stranded DNA is going to replicate, the double-stranded DNA will create, how it will create the copy of itself, what kind of enzymes are involved, at what step this process will take place, where this process will take place, whether it will take place in nucleus or in cytoplasm. So, okay. So we are going to study in this chapter in the later on stages. First, you should have an idea that these are the processes that are undergoing by the cell, right? So this replication is also known as semi-conservative. Why semi-conservative? The semi means, it means somewhat amount is conservative, right? Somewhat amount will remains the same. So what happens during DNA replication? Why it is called semi-conservative? Because these two strands will get separated during the DNA replication. Okay. And the single strand from this, suppose if we call this a first strand and this is a second strand. So using this second strand, new strand will be generated, right? And using this first strand, a new strand will be generated using this as a template strand. It means we are creating the copy using the template number 1. Okay. And in the second part, we are creating a copy using the second strand as a template, right? That is why it is called semi-conservative. It means the original strand from the previous DNA will be there in the during the second replication, right? In this way, in each generation, the first generation will have a copy of first DNA. Okay, in the second generation, it will have a copy of first generation DNA, right? See, this slide gives you an overall idea of what kind of enzymes are involved in DNA replication, right? Key enzymes are DNA helicase, DNA polymerase, DNA ligase, right? What they will do? For example, the DNA helicase that is present, it will unwind the coiled DNA, because for the process of replication to take place, the double stranded DNA needs to be released from the tension, right? It has to be single stranded. So this enzyme DNA helicase will do this part. This will unwind this helix. What will DNA polymerase will do? These polymerase will add nucleotides, right, to synthesize a strand of DNA. What this DNA ligase will do? This ligase will join the fragments. Suppose there is any nick. Suppose there is, absence of nucleotide occurs. So this after the completion of the process, if there is any kind of nick present in the DNA, the newly synthesized DNA, that nick will be joined by DNA ligase. We will study about this in detail, how they are doing these processes, right? So replication in case of prokaryotes and eukaryotes are a bit different, right? Prokaryote replication is rapid and begins as a single origin. It means it starts from a single place, right? The starting point will be one only, right? While in case of eukaryotic DNA, it happens in multiple stage, right? And multiple origin of replication are present, right? See, this DNA is very long strand of DNA. So at multiple points, there will be origin of replication from where the DNA will start replicating. So this is the basic difference between the replication in case of eukaryotes and prokaryotes. Now, after DNA replication, there is a process known as transcription. What will happen in this process? This DNA is double-stranded. Okay. This DNA will get unstranded. The helicase will be, get single-stranded. It will uncover. And this synthesis of RNA will take place. Okay. Of course, for this process, a different kind of enzyme will be involved, right? So in every process, like in replication, in transcription, in translation, we have different set of enzymes. A set of enzyme will work in replication will be different from the set of enzymes which are working in the transcription process. So for each particular step, we will have a particular enzyme to carry out that process. Okay, so this information in the DNA will now be converted into an RNA. And this RNA will ultimately, the information that this RNA is carrying will ultimately lead to the formation of polypeptide chains. That means it will be lead to the formation of protein. So in case of rRNA also, in case of RNA, in case of the process of transcription, three types of RNA are involved. This mRNA, known as messenger RNA, it carries the genetic code, right? tRNA will brings amino acid. It will work as a molecular vehicle. It will go to and fro and carry the amino acid, right? And this rRNA forms the ribosomal structure and will catalyze the bond formation, right? So I already said each and every enzyme will perform a different function. So this mRNA will carry the information. This tRNA will go here and there and bring the particular type of amino acid according to the code. And this rRNA, what it will do, it will join the two amino acids. It will join and form the peptide bond by combining two amino acids, right? Okay. So just this genetic code is nothing but the information in the form of codon. And this codon is what? This codon is nothing but the the nitrogenous base that we were talking about, that are found in the form of triplet, right? So for a particular codon, there will be a particular amino acid, right? So what is happening? See, the DNA is having some kind of information. The RNA is going to, carry that information in the form of RNA transcript. According to that RNA transcript, the rRNA and tRNA and mRNA is going to work. They will, form the peptide. They will join amino acid. This amino acid will, coil and it will form primary structure, secondary structure, tertiary structure, ultimately leading to the formation of a protein, right? So this mRNA, what I told you earlier, will guides the sequence amino acids, right? rRNA will bring ribosomes, read codons, join the amino acid. tRNA, I already told you, act like a molecular vehicle. It's like a vehicle. It is transferring the amino acids in the system. So this is the process. See here, tRNA is bringing the amino acid, right? This is mRNA. And this is the complex structure of rRNA along with the ribosomal unit, right? So after the process of protein synthesis, okay, the some process are also taking place known as post-transcriptional modification. Okay, this is what, see, this primary RNA that is generated through the DNA, the RNA application with carrying while carrying the information, decoding the information from the DNA, this primary RNA will go a set of processes such as capping, splicing, tailing. This will all lead to formation of mature mRNA. That will ultimately lead to the formation of translation. So it is an additional process that the primary RNA is undergoing. It's not like that it will go and for directly synthesize the by through complementary base pairing, the RNA is synthesized and this RNA will be directly used in the process of translation. No, before going into the process of translation, this RNA, this primary RNA will undergo various set of processes which are capping, slicing and tailing that we will read when we are going to study this process in detail. Okay, and that mature RNA, then that mature RNA is ready for translation, right? Okay, so this chapter is going to tell you a lot about what are the things, what are the ways, what are the processes that are involved at the molecular level, right? How is this process is actually happening? What kind of enzyme is doing what process? How the information is going to transfer from DNA to RNA to protein synthesis, ultimately leading to the protein synthesis. Because you already know protein is the building block of our body. For every process, protein is needed. So for each and every process, there is requirement of protein, and information for that particular protein is carried out by DNA through mRNA, right? So this is an overview of this chapter. We will go in detail and study how the DNA is replicating, how this process of transcription and translation is going to take place in a very step-by manner, right? So hope I'm clear. If you have any query, any doubt, you can contact me through my Urban Pro profile. When we are going to meet in the next session, be ready, go through your NCRT, collect whatever information you can collect at the first reading and come back and we will continue this chapter. Okay. Till then, take care. Thank you.